Okay, so here's the Lab 7 review. This is the one we did right after the exam on photosynthesis. So one of the basic things to remember is that, just that basic equation. So everything that goes in to this reaction at first includes H2O, CO2, because remember that's what we're gonna make the glucose out of, and light. So this is gonna excite electrons. Okay, and that's going to go to glucose, which is that big like carbon molecule, and it's going to produce O2, that oxygen, as a byproduct. Now, if you get more complicated than this, which is kind of what we were measuring, we had to remember that this oxidation reaction happens. Or this, actually this reduction reaction, because it gains that electron and gets rid of that plus because the negative comes in. Okay, so in this reaction, we mimicked this reaction with a chemical called DPIP. And as this reaction occurred, DPIP actually would become colorless DPIP-H. So we could mimic this reaction up here using this chemical, and we can use that because this was excited, I think, at 635. So we were able to see that reduce and go away, that blue signal, so long as this reaction was happening. This was kind of our signal that the first reaction where you got, like where photosynthesis occurred in light, you got that first one. So I definitely recommend looking at the preview for this lab because that was actually probably had a lot more details. So time over absorbance of our DPIP. So we start with colorful DPIP at our first time period, right? Now, if we had the treatment that was fresh chloroplasts and it was in the light, so light could happen, light could have the reaction happen, it went very quickly, right? Like that's a really steep drop in the DPIP because remember, this turns into colorless DPIP-H, so long as photosynthesis is happening. Now, if we start here with DPIP and we're doing the boiled chloroplasts, those ones that were completely messed up, they actually worked a little in lab, but in all cases, if you have boiled, messed up chloroplasts or this reaction was in the dark, you really would never change. You would never oxidize to down here. You would just always have DPIP because no photosynthesis, I don't know, no photosynthesis in dark or without the chloroplasts. So essentially boiling the chloroplasts was a control to make sure like none of this reaction wouldn't happen in the boiled ones. And we saw that in the dark, this reaction didn't happen either. We also can measure, photosynthesis by the levels of CO2 over time. We can measure the rate. So we saw that with that gas machine that we used. So a reaction that is going and photosynthesis is occurring is going to consume a lot of CO2 and it's going to go down quickly over time. Again, a reaction that's not using any CO2, the CO2 that's in there is just going to kind of stick right here. Not going to do very much. Okay, so I would definitely look at that preview video because I kind of go into each photosystem a little more in that one. But for the most part, what you need to know about this lab is the DPIP and its relationship to NADPH and like that oxidation and reduction and all that. And just the basic idea of photosynthesis, what goes in, what comes out, and you know, how we studied that and what we would expect to study in any case, like if something went wrong and what we did, like if we pulled a certain chemical, like would it still work?